Hey guys, Mr. Dumber coming at you again from my basement one more time. This is chapter 12 and 13, DNA and RNA PowerPoint part 3 video screencast. And remember, there will be an accompanying assignment to do a summary of this part 3 video PowerPoint. So get comfortable, and this one's going to be a little shorter than the last one. I know you're probably depressed about that, but make sure that you're still doing your summary. Also, please make sure you're getting your assignments in on time to the best of your ability. If you have late work, please get that in. If you've noticed, a lot of people haven't really been, a lot of teachers have not been marking things late, or they haven't been taking points off. So make sure you're getting your assignments in as soon as you are able. If you've missed an assignment or two, just get it in. We'll take it. Okay. So make sure you're doing what you can. Make sure you're staying safe and healthy, and make sure you're social distancing, obviously. This is difficult for all of us, and we miss you guys, and we hope we'll see you again. Obviously, school's out for the rest of the year, but we still have to continue our education. So please try to bite the bullet and stick with us here to the best of your ability. If you're having any issues at all, please get a hold of me or get a hold of whoever your biology teacher is. Okay. Stay well, stay healthy, stay smart, stay safe. Okay. Let's get into this now. I'm going to put my picture off the screen here, so you don't want to see my mugshot. You want to see the good stuff. We're going to begin with 13.1, which is actually the chapter in your textbook that this corresponds to. Okay, so if you want to get your book handy and any other materials that you have for chapter 13 and a little bit of chapter 12, because this builds on chapter 12. Okay, so go ahead and get your things ready. Maybe take a notebook or a pencil and a piece of paper and start making your summary right now while we do the video. Okay, thanks and stay safe. Okay, let me also say that if you have not yet read and looked over PowerPoint part 1 and part 2 of chapter 12 and 13, DNA and RNA PowerPoint, I would definitely go back and look at that first, at those videos that I made, and also at your chapter 12.2, 12.3 reading packets or summaries or anything you've done in your classes that kind of will get you your foundation that you need for this particular part 3 of the PowerPoint. If not, you're going to be a little bit lost. So very, very quick review of where we've been and where we're headed real quick. So this is the first slide of this PowerPoint. We've been through about 40 or so slides of this. So I'm going to kind of scroll quickly through this and highlight some of the things that we've talked about already. Again, if you didn't see the first 2 parts of the PowerPoint, or you haven't been reading your chapter 12 and doing your chapter 12 homework or Google Classroom work, I would highly suggest doing that before you do this. It'll give you a better foundation. So this business of deoxyribonucleic acid, DNA, and its cousin or helper molecule, RNA, ribonucleic acid. What do these things really do? What really are they? Remember, they're just chemicals. They are chemicals that really put in a certain order or sequence, produce a code that gives us our traits. And we said earlier, and I'm going to keep referring back to this, you guys all know that your DNA leads to your traits. However, it's not that simple. Your DNA actually really is the chemical code written in your nucleus of all your cells that needs to pass its code to a helper chemical called RNA, which will then build proteins. And some of these proteins are used to build things in your body. Other proteins called enzymes, these are very special proteins, control your chemical reactions, which give you your traits. So for instance, I keep saying this, let's say you have a certain genetic code in your DNA, a certain order of your letters, your base pairs, A, T, C, and G, which we went through over in the other PowerPoints, adenine, thymine, guanine, and cytosine. These 4 letters, even though there's only 4 of them, give us an immense amount of diversity because we can put them in a different order for each individual. And these orders can be very, very, very long. The entire sequence of your genome is something like 30 billion pairs or 3 billion pairs, I forget which it is. That gives you a ton of variety, even though we only have 4 chemicals to work with. We can all be very, very different, and we can be different from other species as well. So here's how it goes down. DNA, the order of your chemicals, your A, T, C's, and G's in your DNA will copy to a helper molecule called RNA, which will then, again, select certain parts of proteins. We call them amino acids, and we'll get into that later. But we will build proteins based on the code from your DNA. These proteins, usually enzymes, these enzymes control your chemical reactions, and those chemical reactions give you your traits. So if your DNA has a certain code in it. Let's say you're a brown-eyed person and you got that from your parents, either your mom, your dad, or both. You will have a certain order of your A's, T's, C's, and G's that will then copy to the helper molecule RNA, which will then build this protein enzyme that's very specific based on that original code. That enzyme then would have a code on, I'm sorry, that enzyme would then create a chemical reaction that gives you a pigment that gives you brown eyes, which would be your brown-eyed trait. So again, it's not as simple as DNA leading to your eye color. It goes through all these intermediate steps, and there's some other intermediate steps that we haven't even seen yet, and we will in a little bit. So that's the big picture. And I'm going to show you something else here. If you think about this, big picture stuff, you've probably all seen something like this, right? You've probably all seen the top 10 from the graduating class. So I want you to picture this image here. And this top 10 image was really the result of Miss Hager having all of these kids come down to the library to take this picture, to produce this picture to put in the yearbook or whatever. And not only that, but the kids had to be in the correct order. And if you see this year's top 10, they're in this particular order right here. And these kids did a super job, by the way. But the way this all started was Miss Hager, if you think of her as the DNA, she's the boss. And she stayed in her office, and she's safe in the office, which is like being safe in the nucleus for the DNA. And Miss Hager really had to send a bunch of pink slips or call slips out to get all of these kids out of the building, out of their classrooms, and assemble them all in one place in the correct order in order to produce this product we call the top 10 picture for the yearbook. Keep that in mind. I'm going to come back to that later on, but I just want to give you a little bit of an analogy of how this DNA really works to turn things into protein. So if you picture that analogy, and then you look at this, we said Miss Hager right here has to write a pink slip for each of these kids that she needs to see down in the library to get this picture. And those pink slips have to be taken up to the individual classrooms where these students are so they can pull the kids out of class. And those kids have to be then assembled in the right order, these top 10 students. And then they have to pose for the picture. And the ultimate trait, or really the ultimate product here, is that top 10 picture that can go in the yearbook now. If you look at the analogy here, I've matched these up kind of one-to-one. Miss Hager is just like the DNA. She's the boss, she's in the nucleus, and she stays in the nucleus. However, she needs to get her message out. How is she going to do that? Well, she's going to do that with a pink slip. That pink slip is a special chemical called messenger RNA. Very similar to DNA, but there's some differences we're going to talk about today. How is that pink slip actually going to go get those kids? Well, it's got to be tied to an office runner, paired up with an office runner, or an office page, sometimes they call these people. These are students that, volunteer their time in their study hall or something to go run and get these kids and deliver the call slips or the pink slips. Each kit is really like an amino acid. Now, this is a throwback to chapter 2. If you remember, proteins are built of amino acids, okay? So if the protein is the group, the amino acid is the individual children or the individual students, okay? So the individual students are like the amino acids. The entire group of students in the correct order, and that's very important, in the correct order, are this protein that your DNA is really trying to build. In most cases, the proteins in our body are enzymes. Sometimes there are different kinds of proteins for building structural things like your muscle or your hair or something like that, but oftentimes they are enzymes. And these special proteins called enzymes control your chemical reactions. And the type of enzymes you have are what determine the chemical reactions you have in your body. And of course, all this was determined by the DNA. So keep that in mind. A change in the DNA would ultimately change all of this stuff downstream, which would lead to a change in your trait. So back to this. The top 10 students need to be put in order in the library, right? And that is going to be the protein. I think of that as the word protein with an M at the end, like a team of kids in the right order. Those kids will then do what they need to do, which in this case, the actual action that these kids will do is pose for the picture. That would be like a chemical reaction. What's actually going to take place? The ultimate result of this entire process was to get this picture for the yearbook of the top 10 kids. And in your body, or in any other organism's body, this isn't just humans, this is all living things, the ultimate goal is to build some kind of trait, like we discussed earlier, brown eyes or something, okay? Another important thing to remember is that some of this process is occurring in the nucleus. Some of this process is occurring out in the cytoplasm, which is anything outside of the nucleus. And more specifically, proteins are built on organelles called ribosomes. I'm sure you guys remember those from chapter 7. So Miss Hager is in the office, but she gets her code or her master plan out to the rest of the building, the classrooms, and the kids all are assembled in the library to take that picture. If you look back at this picture, these kids are all in order and in the library to get this photograph. And of course, it all started with Miss Hager's master plan. She was the one who had the plan to get these kids in order to get this picture. The one thing that's not kind of reality in my analogy here is Miss Hager, if she's the DNA, she wouldn't actually be in the picture. She'd be back in the office because the DNA never leaves the nucleus. So I hope you understand this little analogy. It'll become more clear as we go on, but keep in mind, it's a process to go from what the master plan is to what the ultimate product that we build is, okay? All right, so we've been through quite a few things: the discovery of DNA, how we determined which chemical it was that was passing on information from one generation to the next, and how that information was actually leading to our traits. So again, I cannot stress enough, go back and make sure you do your 12.1, 12.2, 12.3 stuff before you jump into this. We also talked about if you can change DNA, you can change traits. And here's kind of a crazy example of that, where scientists took a trait from or really a genetic code from a lightning bug and put it into a mushroom, and now that mushroom will glow like a lightning bug. So they took the specific gene that's responsible for the chemical reaction that allows lightning bugs to glow, and lo and behold, they put it into a mushroom, and now the mushroom has that same instruction, and so the mushroom has that same trait and is able to glow in the dark, which is crazy, but it's kind of neat. We got to be careful with what we do with this, because you can understand that people could do crazy things with this that would be unethical or not the right thing to do. So keep that in mind, but keep in mind also that we could solve a lot of problems with being able to change DNA and fix the genetic code, which would fix diseases on the very, very basic instructional level, which is kind of neat. Then we talked about these scientists. Again, this is old stuff, and how they discovered that the DNA was a double helix. Remember, that's like a twisted ladder, and that there's 4 chemicals that make up DNA: thymine, adenine, guanine, and cytosine. And we remember from the PowerPoint 2 and even PowerPoint Part 1, adenine always bonds with thymine. So if you look here, you always see a green with a red, and guanine always bonds with cytosine. We always see a purple with a yellow. And in my class, we say apples grow on trees, cows eat grass. In other words, A goes with T, C goes with G. And this is, again, more old stuff. We looked at the base pairing principles, and this is very important, because if you know that C always goes with G and A always goes with T across the ladder from each other, if you know the right side, you can easily build a new left side. If you know the left side, you can easily build a new right side, which is what allows DNA to replicate or copy, which is very important to keep our species going. If you can't pass your information on, you can't really survive, and your species would go extinct. So it's very, very important that DNA is able to copy itself. That's truly how we pass our traits down. Remember, we don't pass our traits down, we pass our instructions down. And our offspring or babies build those traits based on the instructions that we give them. Then we talked about a little bit of the structure or the makeup of DNA. Remember, DNA is built of DNA is built of a subunit called a nucleotide. And a nucleotide has 3 parts. It's always got a 5-carbon sugar called deoxyribose. By the way, that's the D in DNA. It's always got a phosphate group, and it's also got one of those 4 bases, A, T, C, or G. So here's a typical nucleotide. This pentagon shape here is the ribose, the deoxyribose sugar. This is the phosphate group over here, and over here would be one of those bases, A, T, C, or G. Keep that in mind as we go through RNA now, because it's also built of nucleotides. And again, more review stuff. adenine, guanine, thymine, cytosine. These are the letters that make up our genetic alphabet. Of course, they're not really letters, they're chemicals, but they code or give instructions for certain enzymes, certain proteins, okay? And again, double helix stuff, right? A always pairs with T, apples and trees. Cows always eat grass, G and C, right? We did the structure of DNA. We said DNA is much like a two-lane road with one lane going maybe southbound and one lane going northbound. But no matter what you look at here, no matter which one you look at, which side you look at, you can always predict what the other side is. Again, that becomes very important for being able to copy DNA. If I have a T here, a tree, I'm going to have an apple and A across from it. If I have a cow here, I'm going to have grass across from it. So this is very important in being able to replicate DNA. If I know the left side, I can build a new right side. If I know the right side, I can build a new left side. Double helix is the structure. Here are some of the people that helped discover that. Again, this is all old stuff. Rosalind Franklin, who really took that first picture called Photo 51, that discovered that DNA was a double helix at the microscopic level. This is even smaller than microscopic, really. This is almost nanoscopic. This is very, very, very tiny stuff, smaller than obviously a nucleus, smaller than obviously a cell. This is at the chemical or molecular level. This is super, super tiny stuff. All right, Watson and Crick came along and they helped discover, based on the information they learned from Rosalind Franklin's picture here, they helped learn that DNA was in fact a double helix, a twisted double ladder, almost like two spiral staircases kind of wrapping or twining around each other. All right. And we kept going with this. Edwin Chargaff predicted, or actually predicted, and then actually observed in his experiments that the amount of adenine he found in DNA was always equal to the amount of thymine. The amount of guanine was always equal to the amount of cytosine. And so we came to conclude that A must always go with T, G must always go with C. Peanut butter always goes with jelly, ham always goes with cheese, or if you like, again, apples and trees, cows and grass. All right, Chargaff's rules. Went through the differences between eukaryotic and prokaryotic DNA. Of course, prokaryotes don't have a nucleus, so their DNA is kind of floating loosely in the cytoplasm. In other words, they have a mayor, but he doesn't have a city hall. Eukaryotic cells, they have a protective structure around their DNA called a nucleus, and really it's just like a city hall for the mayor. So eukaryotes have a mayor who's in city hall. Prokaryotes also have a mayor, but he doesn't have a city hall. In other words, DNA is protected in eukaryotes. DNA exists in prokaryotes, but it is just floating around loosely in the cells. All right. And if you forgot about that stuff, please go back and review it so that we can get to the new stuff, the good stuff today. We talked a little bit about DNA replication. Remember, replicate means copy. And copying is necessary because if you can't copy your instructions, you can't give a copy to your offspring because you need a copy for you and your offspring need a copy in order to produce their own traits. So you can't just give your offspring your DNA because then you wouldn't have your own instructions. So you have to be able to copy these instructions for every cell and for every baby that you make. So it's important that DNA can copy itself, and it's really a beautifully simple process. Because DNA always follows base pairing laws or rules, A always goes with T, C always goes with G, it's actually pretty simple to see that if I know the left side, I can make a new right. And if I make a right side, or I'm sorry, if I know the old right side, I can make a new left. We call this semi-conservative replication. So each replicated double helix is half original, in other words, half old, and it's half new. Let's look at this a little bit deeper. We were on this part last time. This is where we ended last time, and we're going to begin today's real new material. So DNA replication. If you look at this slide, DNA is a double helix. When it needs to copy itself, it unzips right here. It's like a fork in the road. It splits, right? The old left side can easily make a new right side. So the old left side is shown here in blue. The new right side is shown here in green. The old right side, shown here in blue, can easily make a new left side, shown here in green. So now we will go from having this one double twisted ladder, this one double helix, to having two identical copies of that double helix. One of these will end up in the original cell that made it, and the other will be able to be passed on to a new cell so that that cell will have instructions as well. Notice these are both going to be identical. However, there could be mistakes made in here, and those mistakes we call mutations. When those mutations occur, the instructions are not what they were supposed to be, and sometimes that leads to problems. Sometimes that actually could lead to something good, believe it or not, a good mistake, and sometimes they really don't have much of an effect. Think about if you're writing a paper and you have a couple spelling errors, it might not affect anything, or it might affect it really, really severely. So it really depends on what we're talking about and where the mistake was. Okay, so for today, this is going to be RNA. This is all the new stuff, okay? Notice RNA, it's not DNA, it's RNA. It is not deoxyribonucleic acid, it is ribonucleic acid. And let's take a look at some of the differences between DNA and RNA.
Sometimes it feels like DNA gets all the credit. Yes, the structure of DNA is amazing. It has a beautiful double helix structure, a twisted ladder, it has its own emoji. DNA stores genetic information and codes for your traits. However, sometimes what gets left out is how important RNA is. Without RNA, you actually couldn't get that genetic message out to your cells so that they can start producing proteins. We talk about this in protein synthesis. RNA is a very important biomolecule, just as important as DNA. In fact, RNA is even hypothesized as coming first, before DNA, in the RNA world hypothesis, something we need to make a separate video about. So let's compare and contrast.
That was interesting what they just mentioned about RNA maybe coming first in the world before DNA as far as when life evolved. We have a lot of evidence for this. And by the way, many viruses have only RNA. They don't have DNA. They just have RNA, and they still get the job done. So that's kind of interesting.
DNA with RNA. First, you will find DNA and RNA in all living organisms. In eukaryotic cells, DNA tends to be found in the nucleus, while you can find RNA both inside and outside of the nucleus. Prokaryotic cells don't have a nucleus. Both DNA and RNA are nucleic acids, which are a type of biomolecule. Nucleic acids have a monomer, which, if you remember from our biomolecules video, a monomer is a building block. The monomer for nucleic acids is a nucleotide, so both DNA and RNA have nucleotides. The nucleotides of both RNA and DNA have three parts: a phosphate, sugar, and a base. Let's draw 6 nucleotides of DNA and 6 nucleotides of RNA. DNA is generally double-stranded, and if focusing on these 2 strands of nucleotides here, you can see they run anti-parallel to each other. RNA is generally single-stranded, so you are just seeing one strand here. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. This makes sense because DNA stands for deoxyribonucleic acid. That's helpful to know because the deoxyribose is a sugar, and nucleic acid is that type of biomolecule it is. RNA stands for ribonucleic acid, as its sugar is ribose.
Keep in mind these differences, please, between DNA and RNA. So far, we've talked about DNA is double-stranded, like a double-twisted ladder. RNA is single-stranded, so like a very, just like one spiral staircase on its own. Please also keep in mind the sugar, or this pentagon-looking structure in DNA, is called deoxyribose, which puts the D in DNA, and the one in RNA is called just ribonucleic acid. You're going to learn something else. Instead of A, T, C, and G in RNA, you know that A, T, C, and G are in DNA, but in RNA there is a substitute chemical. RNA has adenine, it has guanine, it has cytosine. However, thymine is not found in RNA. Instead, it has a substitute molecule or chemical called uracil, and you're going to see that coming up here. Again, keep in mind differences between DNA and RNA.
The bases in DNA are adenine, thymine, guanine, and cytosine. It helps to remember the popular mnemonic apples in the tree. That helps you remember that A goes with T, and car in the garage, so C goes with G, to understand how DNA bases pair. The bases in RNA are adenine, uracil, guanine, and cytosine.
Again, look at that difference. RNA single-stranded, and RNA, instead of having adenine, thymine, guanine, and cytosine, we have adenine uracil. That is a huge concept of this whole part 3 PowerPoint. Please understand, RNA is a helper molecule to DNA, but it does not have A, T, C, and G. It has A, U, C, and G.
Notice the different one? It's uracil. So you have to change that popular mnemonic device here. Instead of apples in the tree, maybe the apples are under. Car in the garage still works though. We mentioned earlier that DNA codes for your traits, but it couldn't do that without RNA's help. In our protein synthesis video, we talk about 3 different types of RNA and their very important roles. mRNA, which stands for messenger RNA, mRNA's job is to carry a message based off of the DNA. In eukaryotic cells, DNA generally stays in the nucleus, but mRNA has the ability to leave the nucleus to take the message.
Remember, we talked about Miss Hager being like the DNA who's in the office or the nucleus, and we talked about RNA, or at least one type of RNA, being like the pink slip or the office slip that she writes to go have the kid paged out of his classroom, right? Now there's going to be a separate RNA called transfer RNA that actually takes the message or the pink slip to the classroom. But in this case, in the analogy, it's the nucleus where the DNA is transferring its message to messenger RNA, which is then going to transfer its message through transfer RNA, and that's going to take place on a structure called the ribosome. Remember, this is one of those organelles inside of cells where protein is manufactured or made. We call that protein synthesis. This is where protein is going to be synthesized or made.
To a ribosome. Ribosomes make protein, and RNA is actually a major component of ribosomes. This type of RNA is called rRNA, which stands for ribosomal RNA. Finally, we discussed transfer RNA, or abbreviated tRNA. Its job is to transfer amino acids to match the correct mRNA codon.
Think about this as the office page that is carrying the pink slip and going and picking up this amino acid, which is the child that it had to get from each classroom. So this is one of the top 10 kids, and it's going to match up with the different pink slips to make sure that these kids are assembled in the correct order in the library to get that picture of the top 10. Again, keep thinking back to that referral or that analogy to the top 10.
Codon charts using mRNA codons have been developed so that you can actually see which amino acid is brought for each mRNA codon. When those amino acids are joined together, they make a polypeptide chain. Proteins are made of one or more of these polypeptide chains, and proteins have tons of different roles, but we don't want to spoil it. Check out more in our protein synthesis video. Before we go, let's try a little 3-question quiz, shall we? Just pause it after the question so that you have time to think about it. Question 1. If I have 8 DNA nucleotides, how many DNA bases do I have? And also, how many base pairs? The answer: each nucleotide, regardless of whether it's a DNA or RNA nucleotide, has a phosphate, sugar, and a base. So 8 DNA nucleotides would have 8 bases. DNA bases pair like this, and that's 4 DNA base pairs. Question 2. If one strand of DNA has these bases shown here, ATTGAC, can you complete what the complementary DNA bases would be for the other DNA strand?
Okay, keep in mind this is DNA. So if you know the left side, ATTGAC, and by the way, these could be in any order depending on who you are and what your genes are, right? Up and down, we can have any order we inherit. However, whatever's on the left side dictates or determines what's on the right side. So see if you can fill this in. What would the other side be? If this is my left side, what would my right side be? Also notice, if I have an A here and a T here and a T here, I don't have to have an A and a T matching up and down, but I have to have an A matching with a T side to side. Again, any order is possible up and down, but whatever the up and down order is of the left side, it determines or dictates or locks in what's going to be on the right side. So see if you can predict what this right side of the DNA molecule would be.
The answer. So remember those base pairing rules for DNA. and the popular mnemonic. If placing the bases in this image, reading in the direction shown on this image, they'd be TAACTG. Question 3. In a process known as transcription, a complementary RNA strand called messenger RNA has to complement the DNA. More about that in our protein synthesis video.